I'm Dr. DeBusk, and in this video I'll talk about the communication within the plant. Most responses occur in tissues or organs different from those that sense the stimuli. In root caps, certain cells called statocytes have large starch granules or statoliths that sink in response to gravity. Statoliths are too dense to float in cytoplasm and always settle to the bottom of the cell, thereby distinguishing down from up. The site of perception is tentatively assumed to be the same as the site of transduction. Presentation time is the length of time the stimulus must be present for the perceptive cells to react in complete conduction. Threshold is the level of stimulus that must be present during the presentation time to cause perception and transduction. In venous flytraps, the threshold for stimulating leaf closure is moderate. The trigger hairs must be firmly bent. This is advantageous in preventing wind or rain from triggering trap closure. The moderate threshold almost guarantees that the trap contains an insect every time it closes. All or none response occurs after the threshold and presentation time requirements are met. For example, many species are induced to flower by environmental conditions. After the minimal threshold and presentation time requirements are met, the plants fully flower, limited only by their general health, rigor, and nutrient reserves. Examples are poinsettia, chrysanthemum, hibiscus, and oats. In dosage-dependent response, the amount or duration of the stimulus affects the amount or duration of the response. In species of this type, individuals that receive only minimum stimulation flower poorly, even if the plant is quite healthy. Examples include turnip, cannabis, and some varieties of cotton and potato. Almost all plant communication is done by transport of hormones. Hormones are organic chemicals produced in one part of the plant and transported to other parts, where they initiate a response. Hormones act at very low concentrations. They appear to be released into general circulation and are not carried specifically to the target. Many regions that are not target regions are exposed to the hormone but do not respond because they do not have the proper receptor molecules. At the site of response, receptor molecules, usually in the plasma membrane, trigger a response when hormones bind to them. Auxin, cytokinin, gibberellin, abscisic acid, and ethylene are the most known hormones, which will be discussed later in the video. Other compounds show hormone-like activity. They are either not present in all plants or are much less well studied. Brasinosteroids are complex chemicals involved in leaf morphogenesis, root and stem growth, and vascular differentiation. Jasmonic acid is involved in defense against animals and fungi. Salicylic acid, related to aspirin, is involved in resistance to pathogens, especially viruses. Auxin is often known as the growth hormone since it stimulates roots to grow in cuttings, as well as many other functions. Auxin, later identified as indole-acetic acid, IAA, is produced as shoot apical meristems grow and initiate new cells. Auxin flows downward, surrounds all stem cells, causing three unique responses. First, auxin triggers cell elongation in the cells of young internodes just below the apical meristem. After stimulation by auxin binding, the plasma membrane pumps protons from the cytoplasm into the wall, weakening it and allowing trigger pressure to stretch it. At lower internodes, auxin doesn't cause any response in fully grown mature cells. They apparently lack the proper auxin receptors. Second, apically produced auxin induces dormancy in axillary buds. Each shoot tip gets only one active meristem. This is called apical dominance. As the terminal shoot apical meristem grows away from the axillary bud, the concentration of the auxin around the axillary bud gradually decreases. At some point, it drops below the threshold. Inhibition of growth is lost. The axillary bud becomes active and grows out as a branch or flower. This twig of viburnum was pruned 2 months ago before this photo was taken. Axillary buds had been completely inhibited by the shoot apical meristem at the time of pruning, but then both became active once pruning stopped the flow of auxin from the shoot apex. The third side of response to auxin produced in shoot tips is the vascular cambium. The response is cell division and morphogenesis. In springtime, buds become active and produce auxin. Auxin activates the dormant vascular cambium and stimulates cambial cells to begin mitosis and cytokinesis in the springtime. It also causes new daughter cells to differentiate into xylem. In some species, apical dominance may involve only the presence or absence of auxin. In others, there is an interplay of 2 or 3 hormones. Active roots synthesize cytokinins. Cytokinins are transported to the shoot and stimulate axillary buds. The ratio of cytokinin to auxin determines whether buds become active or remain dormant. A second interaction of hormones occurs in the vascular cambium. Auxin alone activates the cambium and elicits differentiation of xylem. Gibberellum, also called gibberellic acid, is also present in a healthy stem and causes some of the new cells to differentiate into phloem. Gibberellins can also induce flowering in long day plants if sprayed on them. Whereas normal growth of shoots and roots results in large flows of auxin and cytokinin respectively, environmental factors also influence hormone concentrations. Hormones communicate to various plant parts. A healthy leaf produces and transports enough auxin to suppress activity in the abscission zone. A damaged leaf produces less auxin, insufficient to prevent abscission. Autumn stimuli may cause the production of ethylene, which then suppresses auxin synthesis and transport. Premature abscission of fruits is prevented by the presence and export of auxin through the pedicel. Auxin causes the developing fruits to release ethylene. At maturity, the high concentration of ethylene stimulates the pedicel abscission zone, overriding the auxin concentration. Bananas produce large amounts of ethylene as they ripen. This diffuses away and stimulates other types of fruit to ripen rapidly. People often place a single banana or just even a banana peel into a plastic bag with other fruits. The plastic traps the ethylene, which then causes the other fruits to ripen faster and more fully. Similarly, a bruised apple produces large amounts of ethylene, which causes other apples to overripen. Thus the old saying, "One bad apple spoils the barrel." Light is the stimulus in phototropism. Therefore, a pigment is needed and its absorption spectrum must match the action spectrum. Blue is the most effective wavelength for phototropism. The pigment whose absorption spectrum matches that is a small protein called phototropin. The site of perception of blue light is the stem tip in some plants and the coleoptile, the outermost protective leaf at the stem tip, in others. When illuminated from directly above, oat seedlings grow upward. When an oat seedling is exposed to light from one side, its outermost sheathing leaf, the coleoptile, bends and grows towards the light. If the coleoptile apex is covered or cut away, no response occurs to unilateral illumination, and thus the tip is the site of perception. In dark conditions or overhead lighting, auxin is transported symmetrically down the coleoptile, causing equal amounts of growth everywhere. With unilateral illumination, auxin is redistributed, with the darker side transporting more auxin than the lighted side, so the darker side grows faster, resulting in curvature. Auxin can be collected in small blocks of agar or other absorptive material, then placed asymmetrically on a decapitated coleoptile. The side receiving auxin grows, but the other side does not. In positive gravitropism in roots, the root cap acts as the organ of perception. After the lower side of the root is detected, a growth inhibitor is transported to that lower side of the root cap and then into the root. It slows growth on the lower side. Root growth is opposite from stem growth when it comes to the amount of auxin to produce rapid growth. Almost all plants must reach an adult phase before they can be induced to flower. In the juvenile phase, plants are incapable of flowering. Conversion from juvenile to adult is called phase change. Cold temperatures stimulate phase change in biennial plants. We know little of what stimulates phase change in perennial plants. The green base of this melocactus is its juvenile phase, and the red top is its adult phase. The two are parts of a single shoot produced by just one apical meristem. While young, the plant grew for years as a juvenile, with a body resembling that of many cacti, but it was unable to flower. Once old enough, it underwent phase change, and the apical meristem switched to producing the narrower, very spiny adult phase called a cephalium, which can flower. The adult portion cannot photosynthesize because of the dense covering of spines. For the rest of its life, this plant will continue to grow as an adult, with the cephalium growing taller and the juvenile base remaining as it is. No new photosynthetic tissues will be formed. The green parts of this espostoa are its juvenile body, and the brown strips is its adult phase. While young, it grew as just a juvenile that had about 20 branches, but as each branch became old enough, each underwent a phase change. Only a strip of each branch became adult, however. The rest of each branch continued to develop with juvenile characters. Flowers are produced only from the brown adult regions. Unlike melocactus, new photosynthetic tissues are produced every year. In some annual species, size is the only important factor in the induction to flower. In many species, photoperiod day length triggers the transition to the flowering condition. Short day plants bloom when the days are short, spring or fall. Long day plants bloom when the days are long, in summer. Day neutral plants do not respond to day length. To measure day length, the pigment phytochrome detects the presence or absence of light. Night length is actually the critical factor, so a long day plant is in reality a short day plant. Each species has a critical night length. If a short night plant receives nights shorter than this critical length, it flowers. This is seen in this experiment where both long day and night are simulated, but only the short day long night plant blooms. A long night plant needs nights longer than its critical night length to flower. Presentation time varies considerably. In some morning glories, one photoperiod of proper length induces flowering, whereas at least 1 or 2 weeks of proper photoperiods are necessary for other species. The accuracy of time also varies. The most accurate species is henbane. It must have nights shorter than 13 hours 40 minutes. Photoperiodic control of flowering and other processes, such as the initiation or breaking of dormancy, is more common at locations farther from the equator. The sites of perception for night length are young leaves. The site of perception is not the site of response, so a chemical messenger must be transmitted between the leaf and the apical meristem. Plants contain endogenous rhythms. Certain aspects of their metabolism cycle repeatedly between two states. The cycle is controlled by internal factors, by an internal clock. In uniform conditions, the endogenous cycle typically differs slightly from 24 hours. In nature, the rhythm is exactly 24 hours long because light is able to reset the rhythm. If a period is approximately 24 hours long, it is a circadian rhythm. If a short day, long night plant is placed in continuous darkness, it will flower. The plant can be prevented from flowering by giving it red light night breaks. But these are effective only if given at those times of the endogenous rhythm when the plant is expecting darkness. If given when the rhythm is expecting light, the light breaks are ineffective and the plant will bloom. All flowers share the same basic developmental organization. Most flowers have 4 types of appendages: sepals, petals, stamens, and carpels. Variations in presence, absence, and number occur from species to species and even within species. This wild rose has only a few petals and is a single flower. Many cultivated roses have been bred to be double flowers, with a mutation that causes them to produce extra petals in place of stamens. The basic organization of most flowers can be explained by a hypothesis called the ABC model. The ABC model proposes that the outer part of a floral apical meristem is controlled by gene A, the inner part by gene C, and gene B overlaps the two. Notice the outermost area is controlled only by gene A and produces sepals. The innermost is controlled by gene C and produces carpels. Petals are produced by the zone where both A and B are active, and stamens occur where both B and C are active. If a mutation inactivates gene B, then the floral apex has only 2 zones. One controlled by gene A produces 2 whorls of sepals, and the one controlled by gene C produces 2 whorls of carpels. If gene B is inactivated, then the floral apex does not produce either petals or stamens. In conclusion, hopefully you learned a little about how communication occurs within the plant.